Cool. So, to be very clear, this is not a lecture, just a research talk.
It's not even a talk, it's a research meeting.
Right. Okay,
that's
it's
the
a research
difference.
meeting.
That's right.
Right.
Yes,
Okay,
discussion.
cool. So, after now 2 presentations about an oscillatory interference model, it was Burgess, Florian, and Keith paper from 7, and the continuous neural network model, that was the one by Burgess and 2009. I'm now going to present this hybrid model by Bush and Burgess, which sort of put many of these pieces together. Some of them a little bit different than they were originally implemented here, but all the ideas that I presented still hold. And so maybe it helps a little bit to just go take 5 or 6 minutes to quickly go through these to remind ourselves. So first we had this oscillatory interference model, which was this thing here, right, with oscillators that have some frequency that changes with the speed in different directions. And if you superposition all of them, then you get a firing grid out of it. And more than that, you also get theta precession. So such that the firing of the spikes is at first late, and then in the center of the field, it's kind of in line with theta. And then when you are about to exit the field, the last couple of spikes are always very early with respect to some theta signal, right? And the nice thing about that model is that it addresses, for one thing, the dominance of theta, all these oscillations that are recorded. So these are different cells recorded in medial entorhinal cortex. So that's a dominant thing, and theta is more powerful in the whole hippocampal medial entorhinal cortex than really anywhere else. So it's kind of probably does something, right? And then it explains nicely this theta precession, which I showed you this plot for. So this fact that the spike time relative to some theta precesses, and it turns out that precession actually codes for some distance to the center of the place field that you are walking through. So actually there is spatial information in the precise spike timing, not just in the firing rate, as would be indicated by these rate plots that we know, like these heat maps, where you just think, okay, if the firing rate is high in somewhere, in a firing field, no, it's more than that. The precise spike timing can tell you something, how you are relative to that firing field, whether you're far in or far off.
Yeah.
The nice thing about that it also explains some of the other cells, like these band cells, for example, sort of spatial non-grid cells. Right? Most cells in medial entorhinal cortex.
Yeah, was that because of if you didn't make the assumption about the orthogonal dendritic branches, then that's what that's what you'd end up with, right?
So the idea is,
right?
exactly. If you drop one of those oscillators here, right, then you're gonna get sort of band-like.
And that was what struck me as the most improbable thing about this theory. The frequency thing makes a hell of a lot of sense. But that particular component sort of
right.
Could be, just it's a little hard to imagine.
Right. I mean, maybe we can talk a little bit.
Well, we could, but anyway, that was the nice thing about the band cells, because I think we had a little brief discussion hey, could all this other stuff be built up from band cells? But
right.
It seemed like that was good.
Well, at least, I mean, it is true that at least the grid cells are not the majority of these spatial cells that have some spatial preference. Most cells, I think 43% was the number that I got from, was it Moser or Keith? I don't remember. And it's 20 or 30% are grid cells, and 40% are, they call them spatially periodic cells. So
do they say periodic?
Yeah, they say periodic because, of course, there's a frequency component in here, right? Even if it doesn't repeat. I mean, there's sometimes you see these bands that are repeating, like this one, it kind of gets repeated here. You would see that in the autocorrelogram, but even if it's just a band, it's still, there's a spatial periodicity to it, in the sense that there's a frequency component in one direction.
But if you don't have another band, you're saying there's a frequency component?
No, I think they still...
There's always multiple bands, right? Or no?
No, even if it's just a single band, if it's not a border cell, I think they still call it a spatial periodic cell.
Huh.
I'm not exactly sure.
That's weird. That's weird.
But
does that make sense to you, Marcus? Why do they call it a periodic.
What he's saying is if the spatial autocorrelation is anything but just a big blob of nothingness, then that means there is some periodicity.
Oh, I guess I'm having a different understanding of what periodicity means.
Just that implies that if the cell is firing right now, like, I mean, this is a spatial autocorrelogram. The center means like the center's nothing. If the cell's firing right now, the fact that this is red down here means the cell also fires if I step back. Everything's correlated with.
But that would, if you step back, that means it'd be another band, another place would. Right? Yeah.
I mean, this picture implies something similar to it.
Okay, I think I get it. I got it. I'll let it go.
So I think that's what Flo is saying.
Okay. All right.
Anyways, it's just a nice side effect of this model that it also explains some of these other cells, like these band cells. And one of the cool things is it has like a very straightforward learning rule. So like if you want to figure out how to like select from like some random spread of different velocity controlled oscillators in different directions, like a nice set, turns out you want to have like 60 degrees separation, then there's a very straightforward learning rule. You just want to maximize the postsynaptic firing, so the grid cell firing should maximize under exclusion of collinear inputs. So if you can somehow build a learning rule that does like maximize the postsynaptic firing, then you will automatically converge on selecting inputs that are 60 degrees apart.
Is that true? Does that assume that there's 3 branches, or?
No, it can be any number of branches.
Okay.
I mean, this example is shown with 3, but you can do this with 6 or 8.
I forget why that's the case, but we don't need to go into it.
Right.
As I just said, I thought this was one of the most weakest part of the whole theory, and you're saying it's a super rule to get that. And I was like, oh, okay.
Yes, because you can show that the postsynaptic firing maximizes when the different inputs that are summed, right, or actually multiplied, have 60-degree separation.
Okay. All right.
Anyways, so but there's of course also like this this CAN model, which we quickly talked about. I talked about the Burgess model, which has different advantages, namely that it's really good with stability and robustness. Keep in mind this is really a network model, whereas the oscillatory interference one is really a single cell model, and so that is quite good at generating stability. The way that it does that is that these cells have this inhibitory surround, right? Sort of like an inhibitory ring surround. And so even if you don't move at all, you're going to get like this nice spatial grid, and you automatically form these attractors if you have any amount of background excitation, right? The activity is going to clump in these clusters. And then, so that means you get like a coherent scale and an orientation, because of course the grid can only move as a whole because all of these bumps are pushing against each other via that inhibition. So if anything is going to move, the whole thing is going to move. Which also means that these things have very stable phase relations. What do I mean by that? It's the notion that different grid cells, if you have 2 different grid cells, right, and you look at their firing phase relative to some field, right, then that relationship is stable. It's stable across time in a trial. It's stable across trials, right? It's stable across different environments, different days. So there must be something rather particularly about the setup of the circuit that guarantees that phase stability. And that's a really nice property of these network models, because of course the whole network can only move as a whole, and the individual firing fields are an expression of the global activity pattern drifting, right? This bump drifting through the network.
We covered all this last week, right?
Right, we covered all that.
Let's get on to the new stuff.
Right. Okay. The problem with this thing was that it's hard to learn this thing, because in order to move these attractors, you're going to need this highly, dedicated, this highly input-specific, directionally tuned, slightly shifted inhibitory surrounds, and it's very unclear how to get there. And the other problem was that the networks, in order to get stability, to avoid the problems that these networks have with rotating and shifting and stickiness and all these things, the only way to get around that is to build rather large networks, which is kind of a problem if we're thinking about the idea that Jeff would like to do, namely put this thing in a column.
Well, also in kind of light of that tank paper, I keep mentioning where they showed that in the entorhinal cortex, it's not enough cells to do that. So
right. So that those are like these boundary things, right? Where you see that the network orientation drifts over time if you don't do anything, and that the network gets stuck, so like low speeds cannot move the network. And so you need to build really big networks to overcome that. Let's see. So to quickly point out, since we just talked about the weaknesses of the CAN model, we might want to briefly mention also that there's, of course, problems with oscillatory interference 1, 2, namely that it's not really all that stable because you need this intricate theta reset mechanism. This is what you saw here in this diagram, right? So at the point at the center of the firing field, you somehow have to delete all the theta phases and reset them such that they all are now centered, right, on that firing field again, to sort of, keep that grid coherent. And that's a rather, to explain that properly how that would work, they made a proposal that some place cell that could do that. But yeah, they didn't really propose an exact circuit for that.
That was the one problem. The other was that basically it didn't result in it's a single cell model, not a network model, and so you don't see the arrangement of these cells. It wouldn't lead to the arrangement as we know them.
Yeah, like for example, these stable phase relationships that have been observed.
So I think those are the big issues.
And the other things about the cell-to-cell relationship that you don't account for the really dominant tessellate inhibition. Like when you measure these cells, they all there's very little excitation in this interaction between grid cells. In fact, it's mostly very strong tessellate inhibition. And this, of course, very nicely explained here, because you get, in the CAN model, you get out-of-field hyperpolarization because all these cells push back against one another, all this inhibitory surround, right? Cool, all right. One of the last big problems for the oscillatory interference model that I hope to sort of, push out now and kind of delete from the board in a second is this voltage-controlled oscillator law. The problem that these different oscillators need to be like cosine directionally tuned and need to have a burst frequency that somehow is linear with speed. So what do I mean by that? It's these equations that we talked about when I presented that model. These, right, where for one, the dendritic frequency increases with some factor times the speed in that direction. And the other thing is that the speed itself is modulated with a cosine in the preferred direction. So this is like rather like particular kind of a thing, like two concrete requirements. The nice thing is, and I didn't mention this, but I'm going to show it now, that this has actually been shown. This is a very nice paper by Welday, where they showed exactly that. So what they're saying here is that there's like these two concrete very predictions for these theta cells, and these have very specific properties. And so what they did is they just recorded from actually various areas spiking activity when the rodent is running in different directions. And then they looked for longer stretches so that you would have multiple theta cycles in different directions. And then they plotted that out and they found exactly that, namely that cells have a cosine-tuned directional preference and a linear increase in burst frequency with speeds, which are the two requirements that the oscillatory interference model makes. Which is great because it means that that piece is kind of short, so it's not really a weakness anymore, given that this has been shown. So now we put these things together a couple of years later. It's now 2014. And we are noticing that many of the strengths and weaknesses are complementary. So, for example, the CAN model is bad with the extracellular stuff, like the theta stuff, but the oscillatory interference model is good with it. The CAN model has lots of stability, but the oscillatory interference model is bad with it. The CAN model explains only grid cells, but the oscillatory interference model, might explain band cells. So there's lots of different things where these models are complementary, and so that leads to the idea: can't we put them together in a way that preserves the strengths of both models, but eliminates some of the weaknesses.
Is this the first presentation that people who ever suggested this combination? Was this the first one, or was this just a typical paper?
As far as I know, that was the first one that very explicitly proposed. This is the Journal of Neuroscience paper.
Okay, good. All right, that's good.
Right. Okay, good. And so the most important figure.
And you said, I'm sorry, you said it was 2014. 14. Okay. 6 years ago.
Exactly. So as you can see already from all these neuron parameters here, this is a bit of a biophysical model. It still has lots of heuristics in it, which is I'm going to critique it for in certain places. But overall, this model uses conductance-based neural model. So such that the voltage, right, change of a neuron depends on capacitance and some input current, and all the input currents are piped through like some conductances. It abides by this VCO law that I mentioned. The voltage-controlled oscillators are here, so you get the cosine directional tuning, and you get the linear dependence with speed, right? For these oscillators. And this is now the main engineering figure that we're going to need to talk about. So what you see here is the following, right? You have the grid cells in this neat little array here. Actually, each of those circles is not one grid cell, but it's actually 48 grid cells, I think. And then they get inhibitory input from these theta cells.
I'm sorry, wait, you skipped over an important point there, right? The 48 grid cells, are they, why are they grouped together that way? Why do you say that? I mean, what are they all grid cells that are, how they relate to one another, those 48 cells?
Those are grid cells that have the same, implement the same grid pattern.
But different phases?
Yeah, well, they need to get different phased inputs from the VCOs in order to have different.
But you didn't state that. I want to understand what's the difference between, because you have an array of grid cells, and then each of those grid cells, 48 grid cells. So what the hell does that mean?
Each array is 40, each of those black circles is 48 cells that have the same firing fields. That have implement the identical 36 distinct grid patterns.
Right, they're distinct. I'm sorry. They have the same phase, but different. I mean, are they identical? Or are they just like redundancy?
Yeah, there's redundancy. They receive the same inputs.
No, but do they encode the same thing?
Because they receive the same inputs from the VCOs, they actually encode the same things. Yes.
So they're just redundant cells.
Right.
And why do we need redundant cells?
Because it's been observed that there's redundancies in the grids. Like you might find two cells that actually have the same spatial phase.
Yeah, but we're talking ones right next to each other, right? So
no. This says nothing about topology. Well, this is connectivity. Okay. This is not
well, I'm trying to make the distinction between. That's important to understand. I'm looking at this diagram. I need to know how to picture this in the cortex or in the entorhinal cortex.
Yeah, right? It's hard because it's not a topological model.
Well, we need to do it. That's part of our job. So I'm thinking like, I'm going back to that image in the 10th paper, which is very important in my mind. You know, we show these phase clusters, and in the phase clusters, you have cells that represent, you know, they're all the same sort of spatial griddiness, but they represent different points in space. And there aren't 48 cells like that. So the question is, where are those 48 cells? I mean, how do I lay this onto the topology of the brain?
Well, you can imagine that these might be if there's multiple bumps, like you saw in the CAN model, for example.
So each one of those cells, each one of those 48 would be one of the bumps. And then why do I have? And then there's an array of these, and how are they different?
They are different. They implement different grid patterns, right? Because they receive different phased inputs from
different grid patterns. They're like this is like the 1.4 scale or something like that. Is that what we're talking about here?
No, this has nothing to do with scale. This has to do with the different phases. So this implements, right, this connection that you see, right, with voltage-controlled oscillators, 6 different ones, right? They're actually different in like 60-degree increments, right? And they're firing at a certain phase. I think it's denoted here in red, right? So this specific set will implement one specific grid pattern, right? And all of the cells that receive these same inputs will be part of that same grid firing pattern. If you shift,
but that could be like, that could be, so I'm sorry, I'm still confused by this. So is that like one phase cluster in Tank's paper?
I'm not exactly sure this term of phase cluster in the Tank paper.
Well, I've talked about it several times. It's the idea that within a very small section of entorhinal cortex, you see all the different phases of a particular grid pattern.
Yeah, you could think about it that way, yes. So the idea is that this whole array of all of these spans the full phase range. So they talk about it a little bit abstractly as sort of like delta phi.
I'm sorry, I'm dense about this. I'm still struggling. Marcus, can you help me on this?
Well, I have one question. Right now we're using the word phase a lot. Are we always talking about spatial phase?
We're talking about spatial phase.
So yes. All of these five are spatial phases? Or?
So you get a spatial phase in the grid cells, but the way that you get any movement, right, is because of the temporal phase of the voltage controlled oscillators.
Okay, so these phis up top are temporal phases, or
no? The capital phis are directions. These are.
Okay.
I would, if you just let me talk.
No, the problem of this is that you can go. If I don't understand this diagram, I'm going to lose everything.
Right. I would hope to explain some pieces of.
No, but I won't. You already lost me. Okay. So you go further, I will not be following. So I think it's important, at least in my mind, to make this useful. My time is to understand what they're talking about here, because that's a weird diagram. It doesn't look like brains. It doesn't look like neuroscience or anything.
I would love to explain all the elements.
Well, I'm trying to ask for the most basic one. We haven't even gotten past that yet. Right. So and maybe it's terminology, because we're not using it the same way or something like that. So I'm trying to understand. You said there's 48 cells in there. And I don't know what the difference between them is, if there is any difference at all. And at one point you said there isn't a difference, and the other point you said there is a difference. So I'm still struggling with that.
Okay.
I mean, let's go back to that tank drawing. Can I do that?
Right. Yeah.
Right.
Sure.
So he showed. Okay. Here he had this picture like this, and he showed it like this and like this, and this was like one grid cell module, you might call it. And these were phase clusters, because in here there was a complete set of spatial phases in the room that were represented here. So, and each one of these guys had the same replication of each other. So this represented all the different possible phases that you could get. And this is the same ones and the same ones and the same ones and the same ones, right? Right. So there's 6 phase clusters, all basically representing the same thing.
I get it.
All right. Now, I'm trying to understand how this kind of picture relates to that picture.
Okay. If I'm assuming, I don't know, right? When you mean phase cluster, right? Do you posit that this is a single cell?
Yes, he does.
Okay.
He shows that.
All right. If so, then just pool all of these together, and then you get a nice big pool. That is these 48 cells.
Okay. Now, does that? We're going to get to it in a second, but that number seems higher than that picture from Tank, which suggests it's possible. So, is that number going to be critical here or not?
It's useful for robustness of the model to have more cells.
Yeah. All right, but in this picture, what he showed in a real rat, you got 6 of these. Right. And the other thing was, and we'll get to this in a moment, he showed that the phase clusters are not identical because they have, as we've talked before, some of them are going to be more active than others and some sort of encoding scheme. All right, but for now, we're going to say, okay, so one of those big fat circles is all those cells that are not physically next to each other.
They are part of the same grid pattern.
And then the other ones represent what? They represent
all the other phases.
Okay, got it. Okay.
Right? So that's why you have a full array here, right? I mean, in reality, this is obviously just a schematic, right? Yeah. There's 36 distinct grid patterns that they implement, spanning the full, phase range, right? So what you call this cluster. Right?
Yeah. I forget. He may have shown like 10 different ones. He showed
right
here or something.
So they call this like grid field separation delta phi. Right? If it was a linear thing, right, that would be very easy to imagine. It's just all the cells that fire until the same cell repeats its firing field. The inputs to these grid cells are voltage controlled, velocity controlled oscillators. There's a rather peculiar implementation of this in these what are they called? Like these ring attractor circuits. the idea is, like, they quote some paper here that I haven't read, which actually is originally proposed this architecture. But if we look at how they are supposed to work, you see the following. You start off with generating some burst frequency that they call here the firing frequency of the voltage controlled oscillator. And these increase with speed, and they have a preferred direction. That's this capital phi that you see on top here, right? Phi 1, phi 2, phi 3. And you see that this is a very idealized model because they have exactly 6 different ones to span 60-degree directions, right? So this one points right, top left, lower left, right, lower right. And so these are 60 degrees apart. It's worth noticing that also these are 30 times copied each. So there are 30 voltage controlled oscillators. So this block here is actually 30 different blocks. Again, in the interest of robustness and stability, because the argument is if there was only one cell, right, that would do that, then you would have a very sensitive circuit, right? That cell breaks and all hell breaks loose. If you have multiple voltage controlled, velocity controlled oscillators, then you can then you have a spread, and your overall phase stability is better, right? Because you can sort of average. If one of them is off, that doesn't kill the circuit. So and then each of those.
Where do we think those cells are in the brain?
Theta cells. The exact cells that I
where are they?
So these are, I mean, they need to be directionally selective, and they need to be bursting at theta frequencies. And as far as we know, they're inhibitory cells. So they receive head direction input, I guess, from what's that? Periorhinal cortex or parahippocampal cortex. I'm not exactly sure, but it's in the deeper layers of medial entorhinal cortex. They're like close by.
And are these observed?
Yes, they've been recorded. I just talked about them. The Well-Day paper, where they showed exactly that.
Where do we think these might be in the cortex? In the thalamus, the cortex, in between?
That's a cool question. The Well-Day paper actually also records from thalamus. Although I didn't see them like separating out the thalamic cells they reported there, but I think they're actually really good candidates because, of course, these grid cells are stellate cells, and so they receive input from thalamus, obviously.
I mean, maybe this is understood, and I just don't know it. But there's been sort of a mystery in my mind all along about how motion is encoded in the brain. So you, on the one hand, you have to have an efference copy, which reflects a movement that's occurring. And on the other hand, you have to be able to generate behaviors. So if I say, oh, I want to move my finger from, in the cortex, I want to move my finger from one location on my phone to another, I have to move in a certain direction, a certain velocity, a certain distance. So I have to generate those things. And so the idea that motion is encoding in these shifts, the velocity is encoded by the shifts of frequency, and they're directionally sensitive is a very, very compelling idea. When we first heard that in the oscillatory model, I said, oh, that's going to be an answer, because I couldn't figure out how you could do it any other way. I've been thinking about it for years, how you can code motion. So I thought, not so obvious. So I said, well, this is at least a very interesting pursuit, but it's not clear to me still end to end how that happens. Who decides to change those frequencies, and how do they get changed, and where does it begin? We
can talk about.
Is it known about that? I mean, do they have? Is it, or is this just saying, assuming that these things exist?
I can dive deeper into those.
Well, let me ask you about those. Do they know how it's done, or is it in this paper? They just assume they exist, or do they talk about exactly how these come about?
No, this is a very crude model. I mean, these are like.
So they don't speculate where this comes from.
No, they just say that these cells are fire according to this. So it's a simple Wilsonian, and you have,
so just to answer the question, they're just assuming that these things exist under these principles, but it doesn't go beyond that.
Right.
Okay.
They simply argue that like these cells have been shown by Well-Day et al.
Yeah. Well, it's a fascinating question to me because you need to have an end-to-end solution, and it's not. I don't have that in my head.
If you're curious about it, I'm happy to dig up all the papers on these theta cells. where they exist, and
well, it's sort of like, yeah, I think what I want to know is not all the papers and feature cells. You want to know is like, what is this basic transform that goes on that goes between some sort of the need to get from a point to a point, which you can represent by sort of grid cell transform, grid cell operations, into this frequency domain. How does that transformation occur? There's like you're going between a firing pattern or firing pattern into a oscillatory frequency change, and that's not all obvious to me how that happens.
Right.
But, all right, if that's been discovered or proposed, I'd be interested in seeing it. I'm not sure it is, but okay. This paper doesn't go there. This paper just says, we'll assume it's that.
Just has a model for this.
Okay. Okay.
So there's already obviously like some strong constraints on this. Like for example, you might observe that the oscillators that go in opposite directions actually need to have the same phase. That has to do with some of the mathematical stability of this. Each of those voltage controlled oscillator rings there has 6 cells. So there's 6 different phases to the oscillations that are sensitive to one specific direction. Obviously, that means there's a total of 36 different firing patterns, because if the phase of oscillators that go in opposite direction has to be the same, and if each of those rings has six possible phases, then after choosing 2 of those phases, then you already have locked down the phases of the 2 opposing ones, so this one and that one. And the third one is actually constrained because, as we talked about when we talked about the oscillatory interference model, you remember that I drew like these bands, so this is an oscillation in one direction. If I now pick the phase of my second oscillator and I say, okay, this oscillator goes in this direction, then I already know that if I want to have a grid, my third oscillator needs to pass through these crossing points in order to give a proper grid. So the third oscillator pair is actually not free. That's why there's a total of 36 of those things that implement one phase cluster. So to reiterate,
did you say what the colors on those circles mean?
Yes, that's the phase. So I can go through it again.
No.
So
I don't want you to. I'll come back to that. I don't think it's important right now.
Right. Okay. So anyways, so to relate this to your figure of these phase clusters, right, there's a total of 36 of these things, right? And they all get input from these voltage controlled oscillators, but there's only a specific set of those that will implement an actual grid. And so all of those.
What you just said didn't make sense to me.
Okay.
We said that according to this model, there are 48 phase clusters in some sense, got the big circle, right?
No, there's 30.
You said there's 48 cells in each of those big circles.
Oh yeah, okay, right. If you want to think about it that way. Yes, I'm sorry. There's
Okay.
48 phase clusters if a phase cluster indeed only one cell in.
Now you said there's 36 things in there. Does that mean there's 36 different representations of? I mean, so if I looked at the tank paper, he'd show 36 cells in that phase cluster.
If you look at where is the picture of the?
He didn't have it anywhere near that resolution, but
right.
Doesn't mean
Right. It's okay. Let me just pick up this one figure here of the different phases. Where it is, here it is, right? Coherent phases. So in order to span the space between the firing fields, so to go from this red one to this red one, you need to go through all the different spatial phases.
Yeah.
Question is, how many different cells are there from here to there?
Yeah.
Right? This model says there are 6 cells, right? Because these voltage controlled oscillators that
there's 6 cells to go from there to there. From a bump to a bump.
Yeah. Yes. Because
I thought you said there's 36 total, right?
So there are 6 cells around here, right?
Yeah.
It's shown here to be 6 to n, but it's really 6. So there's 6 possible phases to this oscillator.
Yeah.
Right? Imagine thus the activity of this thing keeps going round and around and around and around at a speed that is proportional to the movement speed in the direction to the right, right? And so depending on which cell I pick, I'll get a different temporal phase. Depending on which temporal phase pairs I pick here, I get a different spatial phase pattern in the grid cells. There is a total of, since I can pick 6 cells here and 6 cells here, and this will automatically constrain which cell I have to pick here in order to get a grid. And 3 directions already impose constraints on the other 3 directions because there are opposite pairs. That means there's a total of 36 distinct grid firing patterns I can implement with this model.
Okay.
And each implemented grid firing pattern has 48 cells that participate in that one grid firing pattern. Okay. So there's a total of 36 times 48 grid cells.
Yeah.
Okay, good. The next thing is, of course, that inside every phase cluster, if you want to think about it this way, all of the different cells, right, inhibit each other across the different spatial phases. So these grid cells project to a population of, I think it's 12 inhibitory cells. And these 12 inhibitory cells then have a surrounding inhibition ring that increases inhibition the further you go away from that phase, which is exactly the thing that's the basic building block of the continuous attractor neural network, right? This ring surround, right? So now you have in one figure here the 2 things that 2 of the main components of the 2 different models. The top part is an oscillatory interference model, which, right? So different velocity controlled oscillators impinge on a grid cell. And they integrate the movement through the change in the oscillation. And all of these grid cells share disynaptic inhibition through a shared pool of inhibitory neurons, which are tuned such that cells that have a different spatial phase are inhibited strongly. So if it helps to remind, you can take a look at, where was it? Here, right? So this is essentially this inhibitory surround in the continuous attractor neural networks, right? These bumps push against each other. So cells that have one specific phase, this cell here, has a strong inhibition against all the cells around it that are in a different spatial phase. That is implemented by these inhibitory neurons here. And so they have a tuning of this, they have this parameter, right, this grid field separation, which has an impact on the connection strength of the inhibition. But every grid cell here, so these are 48 cells that implement one specific grid, one specific phase set, right, have a unique inhibitory pool. So that means there's a total of 36 times 12 inhibitory neurons.
So each, say that again.
So this one circle down here is 12 inhibitory neurons.
Yeah.
There are 36 possible grid patterns. So that means since each grid cell pattern has 12 dedicated inhibitory neurons, that there are 36 times 12 inhibitory neurons.
Okay. Yeah.
So in essence,
in our case, in this picture over here, they'd be distributed in the different places. So it's inhibitory neurons.
Exactly.
Yeah.
So that's what makes this this model a little bit hard to understand, is that this thing here is not really topological.
Yeah.
But I hope
No, but I want to keep going back to it. So every time you say a new number, I want to put it onto this picture.
All right. Good.
I think that's important.
All right. So the other thing that you're going to need besides all these inhibitory inputs is, of course, some baseline excitation. And they do 2 different things. So in one of the trials, they simply have a uniform excitatory input profile. So what that means is they pick from a normal distribution with a mean.
Just to make sure that somebody's active.
Yeah,
Okay.
exactly.
Yeah.
So that there is any firing, right? Yeah. And that's the same thing as in the continuous attractor neural network model, right? If you remember that, it's the exact same thing where this is all inhibition, but in order to make anything fire, you're gonna need to have some basic excitation.
Can I just put this on here? This is what I'm thinking when I look at this picture up here. I'm thinking like, okay, if I want to put that picture here in this space cluster, I'm going to have this thing here like this. And then you're going to have the same thing over here. So when they show up there like this one picture and they say, oh yeah, there are 36 of these and there's 48 of each one of these, we're separating that out to like, no, it's going to be a small set dedicated here and a small set dedicated here and so on. It's repeated. But just in reality, it wouldn't be so isolated, right? Because it's going to be continuous in this whole module here. So that's a way of thinking about it. But obviously, these are not isolated units. So there's going to be, these are going to continue across here in some sense. It'll be distributed around this whole thing. But that's what we're talking about here. Okay, that's just how I'm thinking about it as I go through this picture here.
Right, and then of course the reason why these are distant apart is because of the inhibitory.
That's the span of the inhibitory.
Exactly.
Yeah,
right. That is the good.
Yeah.
Right. Yeah. Then I think we have a shared understanding.
Yeah. Now again, they're only talking about 6 of these. He was pretty clear about that, and they got 48, and that's just a matter of robustness and in encoding. But I think we can get away without that. I think there's some assumptions in the model that we don't have to stick with in that case.
All right.
So basically, we're relying on the inhibition for stability. Pretty much it. It's really just providing stability of firing, and then
a little bit more than that, right? Because of course it
provides topology as well with the
and it guarantees that it helps stabilize the phase relationships if there's any drift.
Well, so I said stability.
Right. Yeah. Okay. Right.
So stability and it explains the topology that we see. And I guess the reason that the CAN model required so many more cells, I'm guessing, I don't really understand this intuitively, so many more cells, is that if you're going to rely on it to move the bump, sometimes you need all those cells. If we're not relying on it to move the bump, if we're just relying on the CAN to basically be stable, then you don't have to have a lot of cells. It's not intuitively obvious why that's the case, but to me that's the bottom line here.
Right, this thing here, right? The fact that the cells that move the attractor bump in the CAN model, right, are actually in the sheet, and they need to be perfectly balanced. So you need to have a large number of them, or else, right, you don't get that stability. Right. Okay. Good. So now we have some basic understanding. One thing I want to mention briefly here, that sometimes to account for the idea of a place cell, for putative place cell input, they try out a different excitatory input that is actually spatially selective. But we will get to that a little bit later. So the model has a lot of bells and whistles, as you can see here, right? So there's all kinds of different input currents, AMPA currents, MBA currents, GABA currents. They actually even come with different time constants, GABA A and GABA B, and different MBA time constants.
So they run this as like a neuron simulation type of thing.
Yeah, exactly, right. Yet at the same time, other aspects like these voltage-controlled oscillators are really, very coarse, right? They're just this little equation here.
And there's no network here in some sense.
Right, and there's no topological
network.
Right, no topological network. It's you can try to read it into the connectivity, but it's not specifically implemented.
No.
Now, the other thing that they do, of course, which we remember, one of the requirements of the oscillatory interference model is that for this to work, in order to do all the movement integration through these oscillators, you're going to need to have some theta reset mechanism. They have a rather crude implementation of this, which works very nice.
Isn't that just the place cell thing we're talking about? Wasn't that just like, if I know where I am, I reset the whole thing?
Right. So you could do that either through the place cell, or if you have enough grid cells, then you could do it by the grid cells themselves. Because of course, if you now have a pool of 48 grid cells, that means that if you get any drift in the phase of its inputs, you can use the mean to recorrect the thing. And that's kind of what they do. So when they introduce some phase noise, they have now a mechanism by which they essentially through averaging over these, they can sort of drive the phase of the inputs right back to what they should be.
And I think I said on Wednesday or Monday, whatever it was, that I don't think this is going to be the main reason why you do this. I think it's going to be the place cells that do this. There's a lot of evidence for that. And so maybe some of this is going on too, but I don't think, maybe if they need to get to that 48 number for that reason,
maybe.
You're right. I don't believe that this is the way this is occurring. I believe it's mostly occurring through the place cell interaction. And if that were true, and I need the 48 to get to this mechanism, that's why I explained I don't have 48. I got 6.
When you say like 6, I mostly wonder. I'm not convinced that like this is really a single cell.
It may not be a single cell, but
like given that there's an inhibitory surround, I'm thinking more it might be like a very small cluster.
It could be, but they were recording single cells with optics, and they didn't show multiple cells active. Now, is it possible?
I mean, like imagine that it's just a cluster of 6 cells, then you would have your 36 cells right here.
Yeah, but I don't see the evidence that the tank show didn't show that.
Right.
And if I have I already suspect that this is not going to be the way it's mostly done. It's going to be mostly done through place cells. And if the only reason I needed to get more is to make this mechanism work. Why would I add all that complexity? I'm just saying the more copacetic answer is to go with what the data suggests and the theory suggests, which would be to say that don't have 6 cells here to do that. I can get away with 6.
I mean, like. If we're going to talk about the topology of this, I mean, it's reasonable to assume that the kinds of inputs that this cell gets are shared by cells that are close by.
But I mean, I think what you see in all these situations is it's always a bump, right? There's always cells that there's a center and there's a couple cells nearby that have some activity and it drops off. So these are not single cells that, in a oscillatory fashion jump to the next cell, right? It's a bump. So all I'm just pointing out is, I'm just reiterating, if you don't ignore it or not, if you want to, but I don't believe that, I believe that, and for the evidence I gave earlier, was I can go through again, that most of this resetting of the drift is going to be occurring through place cell interaction. That seems to be in my mind, that's the hypothesis I'm working on. It seems to be more setting the data. But it doesn't mean that this isn't an interesting idea.
Right. So this is what they start with, just an oscillatory interference model. Right? So you get oscillators in different directions. The ones that are, in opposite directions have the same phase. And then by picking 2 of these, the third one is essentially fixed because you get this grid then, right? So that gives you this grid. So this is just an oscillatory interference model, and it does the thing that an oscillatory interference model typically does. So you have on a linear track, right, you have like these repeating firing fields where the mean firing rate goes up. There is a baseline like a theta, and the theta actually diminishes in between the firing fields, and then the spikes right sort of in the middle of this firing field. And if you look at the exact phase of these spikes with respect to the extracellular LFP, which they assume just to be 8 Hertz, I think, then you see that there's precession in there, and there's this modulation of theta power. So this changes a little bit once you make this a hybrid model. So at first here in A, you see it's essentially this would be a little bit like a phase cluster actually, where you see that, so A is without oscillatory input. So this is just the inhibitory surround dynamics, which after a little time will generate a bump of activity. Alternatively, you can switch on oscillatory interference input, which will very quickly establish a bump, right, because of the powerful oscillatory inputs. And then when you do that, you will also see that there's now in the temporal autocorrelation of the spikes, there's a frequency, there's this theta shows up, right? You get a grid, just like you did before, but there are a couple of differences now. For example, in the hybrid model, because there's now an inhibitory surround, you see that there is a disynaptic inhibition in between the firing fields. So in the gray zones here, right, which are between firing fields, you get inhibition, dips down, which it didn't before. And you now get, you still have the sort of theta here and the spikes riding on top of it, and you still have precession, which you see here. Although the precession is now less of a range. So if you compare up here in the oscillatory interference model, the precession goes through a full 360 degrees. Once you add the inhibitory surround, right here you can see it very nicely, right? It goes from 2 pi all the way down to 0, right, as you go through the distance across one grid cycle. And noticeably, there is this difference in the theta power in and out of firing fields. This disappears, like this difference in theta power in and out of firing fields, because now, when the cells are inhibited, they're inhibited by other cells that are also theta modulated. So the theta power doesn't change much anymore now. And so this is very good because it gives us the benefit of the intracellular voltage dynamics that we had in the CAN model. Namely, we can explain the hyperpolarization out of field now, and we can explain that there's no change in theta amplitude just because we're in and out of field. Because if we're in the field, it's driven by these oscillators, and if we're out of the field, it's inhibited by cells that are driven by oscillators. So theta stays in the system.
That's sort of obvious. Can I take a diversion on this for a second here?
Sure.
So one of the things that I've mentioned again many times, it's really puzzling about this empirical observations that they have is, and it's related to what you were just talking about, is they say, oh, okay, we got a bump of activity here and here and here and here and here, and if the animal moves, that bump of activity might move down to here, and it might move down to here, and down to here, and down to here, and down to here, and down to something like that. What they showed consistently, meaning that it wasn't random, it was associated, they showed at any particular point in the world that the actual activity levels of these cells vary significantly. So I'll say that, for example, I'll put a big circle on this one, and a big circle on this one, and a big circle on that one. It wasn't equally spaced. It could have been that one, not this one. Doesn't really matter. The point is that there was a definite difference in some of these were very active, some of these are not very active, and that was consistent with a particular location in the world, in a particular state, perhaps. And so it wasn't a random thing. It was statistically important. And of course, we need to having a grid cell module like this is not sufficient to represent all the things you need to represent. It's not determined. And so it really an interesting question is how would so I think this is a way of encoding additional information on top of the spatial field of firing. And the question is, how could that come about? Now you got these guys essentially all inhibiting nearby neighbors. So there's some sort of surrounding inhibition around these guys, and yet I need to. If one of these guys is not very active and one of them is very active, somehow bumps all move correctly anyway. It's as if this guy's not really firing, but it still acts as if he's firing. You know what I'm saying? It's like the whole CAN model requires that these cells are active and you have this inhibition by synaptic inhibition, which basically, and yet I wouldn't have that in this case. If this cell was basically silent at any point in time, but I have to act as if it's still active. The inhibition has to continue on. So that's a puzzling thing to me. I don't understand how that could work.
I'm not sure what you mean by the ambition has.
Well, I mean, the whole, imagine if we want all these bumps to move around together, they have to be bumps. There have to be activity here. And they're saying that there isn't always activity here. It's as if some of these cells are silent or quiet, even though, and then a moment later, the bump will be in the right place here. So it's not like I have 6 bumps. It's like I have 3 bumps active. Yet the whole network acts as if they're all active. You see the problem?
Yeah, I see the problem.
And the question is an obvious or simple answer to that. And hearing you talk about this is like, well, you know, what's really driving the bumpiness here is the inhibitory cells, right? Right. And so
at least it's stabilizing the bumpiness.
Well, the question is can the inhibitory cells change without being driven by the excitatory cells? I mean, this excitatory cell is not excited, it's not behaving well, it's not active. Can I still somehow theoretically move the bumps around even though there is no bump here? That's the question I'm asking.
Yeah, because you have 2 complementary mechanisms, right? You can move it either with the oscillatory interference or with the can dynamics by having other bumps push on it.
Well, so in this case, I'm saying I don't have the can dynamics here. So what if I want the assumption there's no can dynamics right here?
Then it's still going to move just because the same oscillatory, like the theta cell inputs, right?
Yeah.
Let's call them, you know, VCOs. These are presumably shared.
Yeah.
Meaning, if these bumps reset these thetas correctly, then this will still, you know, move correctly even if it's not participating. Because it's sampling from the same theta cells.
Yeah.
So if the theta correction for these theta cells, right, this phase reset mechanism that they have, right?
No, so obviously the oscillatory part will continue to work. Will it?
Yeah.
Explain. I mean, maybe obvious to you. It's not obvious to me. If this cell is not actually active, okay, doesn't the oscillatory model require that cell to be active?
No. Look, in this diagram, right, these connections here are sampled, right? So you sample from this pool of velocity-controlled oscillators.
Yeah.
And so any individual grid cell here, right? So we are comparing 2 different cells inside this box here now, right? These are the 2 different cells. And one of those is strongly active and the other one's a little bit less. So maybe this one, right, picks slightly more lucky inputs.
But those cells work on the idea that you have this shifted frequency. I'm just trying to keep this straight in my mind.
That the velocity.
We actually doesn't move. It's really just saying, oh, I will still detect these 2 velocities, these 2 different frequencies. Yeah, I think I got it. I'm mixing what's moving. It's actually the oscillatory model doesn't move anything. It just activates a different cell where and therefore I have these cells out here, even if they're not active, they will still get their differential input.
Right, because it's a single cell model.
Yeah.
But the single cell will not be lost if the correction of the theta cells is done by the other cells that also receive it.
So maybe would this be the right way of saying it? In order for this system to work, I have to have the can component active for some period of time, or a large percentage of the time, some percent of the time, to basically train the cells to do the right thing. I mean, they have to differentiate themselves. They have to train to learn which of the different what cells are activated on different oscillations, and the CAN model is going to train them. It's going to say, but now once I've trained the system, I can drop out some of the canniness at any point in time, and it'll still work. But I had to have it there to begin with. I had to have it there. It's in some sense, the default mechanism, you have the CAN model working everywhere, it trains, the system learns. But if I drop out any activity at any point in time, the network will continue to work.
Yeah. If you believe that the network is topological like that, right? Then you're gonna need to have a topological mechanism like CAN to implement that in the first place.
But the simple answer to my question is, I'll state it again, is I had to have CAN to begin with. The CAN is how I train the systems, how I learn where the different cells, give me my topology here. Otherwise I wouldn't have the topology. And once I've done that, I can drop out the candidness by some of these cells not being active at any point in time, and no one's really going to get confused yet. You wouldn't want to do that forever, but because it may unlearn its candidness. But okay, so that's a simple explanation for that. It doesn't explain why some of these are active, more active than others, and that's a really key thing for us. And that may involve other interactions, mini columns and things like that. Okay, that was the answer to my question. It's a simple explanation.
All right, so to reiterate now, so the hybrid model makes a couple of differences now. So we do get the LFP and the theta precession, and well, it's unclear if we get answers now, but I guess theoretically we could. They don't actually implement this learning rule. Because they set up this thing optimally. They have 60 degrees separated different oscillators, and they just make them project, and then they sample perfect from each of those pools. So they didn't implement it, but somebody could implement it.
Can I interrupt again? Mm-hmm. Can you give me I don't have an intuition about the 60-degree separation. You said it. I've heard you say the words. They don't want to do that if you try to maximize the overall firing rate or something like that. I don't understand why that ends up at 60 degrees.
Okay.
So I have a cell. I have some number of dendrite branches. This is the way this was phrased. Is these dendrite branches, which representing a different 60-degree shift or something like that, or 60-degree direction, if I think it was. But I said, what if I have 4 dendrite branches? He said, no problem. It'll still work. I don't understand that. How does that come about?
Here,
this
figure.
I want intuition. I don't want the mathematics behind it.
Okay, so right. Intuition, right? So what they tried here are different unique combinations of preferred directions. So they had a pool with velocity oscillators in, 0 degrees, 10 degrees, 20 degrees, and so on to 350 degrees, right? And they just said, okay, I'm going to pick a random unique combination.
Of the oscillator cells.
Of the different oscillator cells. And they showed that it's fine, if the oscillators are at least 20 degrees apart, you already get a grid. So you don't need perfect 60 degrees for a grid. But the firing rate increases as the difference between the oscillators that you pick approaches 60 degrees, which is the one on the left, which is the perfect one.
Yeah, but what's the intuition behind that? Let's walk.
Right. The intuition behind it is the same as the one that I drew here. So say you start off, right? So, simplified, most simple model, just 3 different oscillators, right? If you pick the first 2 at random, right? So I'm just drawing two lines here, right? Then it's kind of obvious that if the firing only occurs when you pass a certain threshold, that you want your third component to go through the connecting nodes in this grid. So you want this oscillator to be at 60 degrees versus the other ones. Actually,
I could take that. I could distort the whole thing, and they still go through the centers of all those things. Why? If you know what I'm saying?
No, I'm not.
Imagine I draw that, and I just twist it a little bit. So it's not all 60 degrees, but they still go through the center.
Would it be 60 degrees with respect to each other? Right?
Well, would they? I just
the third line feature of these.
What's the big one?
Big black one is.
What if I had like this? I'm just trying to understand it, and I have like this. So these are not at 60 degrees, right? And now I could just still go through the centers like that. It's like so just crossing through the centers of these things doesn't give me 60 degrees.
I can give you intuition for that.
Yeah.
So the intuition is that you're not gonna get firing sort of this. You could think of it as like this is obviously just sort of like the center of that band, right?
Yeah.
So there's a certain amount of space around it.
Yeah. So let me jump ahead here. You're saying basically some of these things will be better represented than others. More activity here maybe than there or something like that. Is that where you're going with that?
No, I was going with a different thing. I was going to argue that in order to maximize the firing, you're going to need to maximize the difference, right? That space there, because how how do I think about it? So instead of thinking of these as activity lines, you can also think of them as non-activity lines.
Yeah.
And in order to maximize activity, you need to minimize the non-activity, which then leads you to creating maximal space.
Yeah.
So it's not good to have lines that are too parallel to each other.
Yeah.
And so if you're gonna pick any lines, you're gonna pick the lines that are most.
I'm stuck in a really basic idea here, so maybe it's starting my. Why is that not sufficient as well? It seems to meet the same criteria.
Yeah, you can build a grid with just two oscillators.
So why is it then? Why do we always have three?
We don't. That's just the ideal case.
But we don't see this kind of structure. We see this kind of structure. So why?
I mean, that is not optimally packed, right? So if you were to have activity in this thing, right, you would waste space, right, in here, right? So that means,
can't I argue the same? I'm wasting space in there. I mean, isn't that, what's the difference?
I'm not sure if I follow. If you're going to have inhibitors around these points, right, then you're automatically going to get another bump here, and this now means you have again a hexagonal grid. The reason when you dump spheres
so
in any basin, you get
it's a simple thing here. It's just the packing.
Optimal spatial packing.
Yeah, but focusing on the inhibitory cells, I can understand it. Focusing on the oscillatory cells doesn't help me. Didn't help me.
Okay.
If I just said, what will I get if I just do think about the inhibitory cells and their effect? I get it right away. Okay, got it. I wouldn't end up like that. It's sphere packing. But it was the fact that you were phrasing it in terms of oscillatory desires. I'm like, I couldn't figure that one out. Like,
well, you still need some threshold of activity, right? At what point do you fire, right? And the way that you make best use of your firing with regard to the space that you have is to choose the spatial packing, which is densest, which is always going to be a triangle.
I got it. As soon as I talk about packing, I get it.
Right.
I just talking about the oscillatory things, I didn't get it. So then,
guys, I just want to make a note about time.
Okay, but I'll go with that. Okay.
I mean, we can look at the geometry.
I then have another very basic idea here. So we show these cells and they say, oh, they draw the picture like this. You have this picture in there. And maybe I'm misunderstanding. I don't really understand this picture. I thought you said earlier that these dendrites are themselves going to be sampling from the oscillatory cells, so that they individually have their own sort of oscillation that are 60 degrees apart.
Right. The different theta cells impinge on that, right?
In
an ideal
case, but these dendrites themselves, just forget about what's impinging upon them. These dendrites themselves have this separation of their internal oscillations.
Well, they don't need to, they get spikes, right? It's a Poissonian process.
Yeah.
So the way that gets implemented in this model is quite simply that you get a Poissonian spike, and the probability of that depends on this firing rate, which depends on this VCO law.
I was just trying, what happens if I had,
so there's no oscillation per se, there's just inputs that arrive at different times, and since they are bursting theta cells,
so how does this work if I have 4 dendrites, or if I have 2?
The exact same. I mean, in this model it works with 6.
I guess I don't understand it, because I had this incorrectly, I'm just confused, this idea that these were somehow representing different 60 degrees out of phase of one another. But if I have 4 of them, I can't do that. Or if I have 2 of them, I can't do that. So what am I missing?
Not much really. I mean, like you can pick, and as we showed in the oscillatory interference one, you can pick any number of oscillators, right? You're gonna get some kind of hexagonal pattern. It's just that the best hexagonal patterns you get if you pick, 3 to 6 oscillators that are 60 degrees apart.
So you're saying by 4 that 2 of these might be the same frequency?
Yeah, that would not, like the way that they talk about that this could be learned is a heavier learning rule that would maximize postsynaptic firing under exclusion of collinear inputs. So they presume that there would be some mechanism to make sure that you don't get the same input twice. Other than that, you're free to sample.
All right, there's a whole bunch of words here. I'm trying to translate those into my question here.
Right.
And
Just pick 4 different oscillations. It doesn't matter. You're going to get a hexagonal grid. It's just not going to be pretty unless there's 60-degree angle difference.
when you say it wouldn't be pretty, it would be that sort of distorted one we're talking about.
Yeah, right. I showed you one with just 20 degrees between oscillators, right?
So let's say we're talking about real biology here, okay? If we assume that our I don't know, these are stellate cells or parental cells, but these cells are if they all had 3 dendrite branches coming off the soma, then that would work out well.
I think biologically they say they have like 4 to 8, and ideally you'd want to have 6, right? Because you want to have an oscillator in every possible 60-degree separable direction, which is 6.
So my point was, I was going to say, I'm not aware of them being very regular. They kind of have a distribution of number of dendrite branches. You said 4 to 8. So does that mean then that I won't have ideal grids? It's say, okay, I can deal with that. It's a little sloppy, but it's okay.
Just because you have 4 to 8 different inputs does not mean you have a grid, because you're going to need to have, for example, a reset mechanism to make sure that when this cell fires, the theta of these resets, right? So this whole oscillatory interference model only works with a reset mechanism, and you only get a very nice strong grid if they're 60 degrees apart, but there is a learning rule to get there.
Yeah, but okay, but let's say I have 4 dendrites. I'm trying to understand what happens in that scenario. What occurs when I have 4?
You get a grid.
But is it an ideal grid or not an ideal grid?
Well, I mean, it's going to look like one of these grids here, right?
Just yes or no, ideal or not ideal?
I don't know what you mean by ideal. It's not going to have maximum firing rate.
Okay, great. So not ideal. So okay, so
but it's going to look like a grid.
Yeah. I got it. I just want to understand.
I mean, I think many of the questions would be helped by just a simple toy model where you can set your own inputs and play around with the firing fields that you get.
Yeah, but I never. That's not sufficient for me. I'm always like, I just need intuition.
Right.
I need the intuition. If I don't have intuition, then I don't understand it. You know, simulation doesn't give me intuition. Okay. It's not in my case. Okay. I'm going to just accept it that in an ideal world we'd have 6, but we don't have 6. But it's going to be okay. It will be a little bit less than ideal. Right. Okay.
It's like, good enough. And the nice thing is, of course, is even if the bumps are not ideal, if you add to like a messy pattern like that, a CAN connectivity, then all of these bumps are going to sharpen. Right? That's in fact what they show in this hybrid model, that the addition of, so this was the original oscillatory interference one, that the addition of the CAN mechanism actually increases the amount of spatial information.
All right, I have another related question. And Marcus, you can maybe help out on this one. So this is all assuming we're trying to represent these 2D spaces. And we know the brain has to represent at least 3D spaces and other things as well. And so there's a lot of question about how it does that. So I guess I'm trying to understand, I mean, there's 2 possibilities here. One is that the mechanism is inherently a 2D space. And then on top of that, there's some tricks that are played to get it to represent 3D, like sort of like this coding scheme I was talking about a moment ago, or some other things. The other is that in some parts of the brain that they're actually representing 3D space, and we're seeing in the rat it's mostly a 2D mechanism because that's basically what the rat lives in most of the time, so that's what it settles on. But if the rat truly was a bat, then it might be a true 3D grid cell space. And the question there how would that change? What can we say about that from this mechanism, and how would that change how we were describing this issues we were just talking about, the 16 degrees of separation and so on? I know you've thought a lot about this. So
right now I'm trying to figure out off the top of my head what would happen with this with 3D, and I can't figure it out right now on the spot.
Okay.
And I want to know the answer to that, but I don't know.
No.
In general, so the solution we have at the moment that I'm still kind of working on the paper on, with the 3D grids involving the projections, the pillars, the fields.
Yeah.
I mean, that might be right, but like the evidence from bats and rats that like you get these messy actual grids, like messy 3D grids that aren't perfectly periodic.
They don't look as good as you would like them to look.
No,
Yeah.
I think 3D we're gonna want something 3D periodic.
Yeah.
And like actual 3D grids. And I'm trying to figure out how to get that to work.
And then the
It
opposite,
might work.
in the opposite direction,
Right.
is that the idea that I've been throwing out every once in a while about 1D grid fields.
Well, here you kind of have those in the sense that that's what the VCOs are. Those are the equivalent of a 1D grid.
Okay, that's an interesting idea. So
like
we've been thinking about the VCOs as sort of like dividing themselves in a plane is what we've been thinking about here. That's how this paper is talking about it, right? So let's go with the idea. That was a very interesting statement you said. Oh, they're like one-dimensional things. Right, yeah, I can see that. So what if I divided them up into 3D space? Like, you know, what would happen? Now, the problem here is I'm not sure that the entorhinal cortex does this, but I think the neocortex does this. So what would occur here? It's very confusing to me. It's not obvious at all what would happen if I took the same model and threw in, you know, VCOs that would span three-dimensional space.
It'll be obvious to me in an hour. I just don't know right now.
Okay.
Because right now we have a 2D canon still. There's still the 2D canon involved here.
Oh yeah, that's a topological neural mechanism.
Right, and I feel like that's going to play into this. It's going to collapse into the pillars thing.
Yeah. I mean that's to me this is a really important thing to figure out. And as I've made arguments before, if I could get this to work in sort of a mini column hypothesis with like 1D type of things, it would be really beautiful. Although I have almost no evidence for it. So I'm asking about that. Well, I know you're doing other things, but if in an hour it comes to you, let me know. Okay. I think it's an interesting problem.
Just to draw your attention to it, it's just interesting here that the path integration is being performed by these VCOs, these data cells, and like the grid cells are they're participating, they're helping error correct, and they're helping the process. But something else is actually doing the path integration.
Oh, that's
which is interesting here.
Yeah.
Grid cells in some ways are just a readout here that are providing error correction and provide a metric, a spatial metric.
Well, not exactly, because of course it feeds back, right? Because the way that these theta cells can do path integration is only because you have a reset mechanism that when the grid cell fires, resets their theta.
Okay, sure.
So in some, it's a unified system. It's not one thing does path integration, the other one is a readout. It's
but the point I think it's a good observation, but also a good observation. That's a very important distinction. That brings a whole nother level of indirection into my thinking about it, which I hadn't really dealt with before. Yeah, so now you kind of separate the problem into two parts. You've got the VCOs, which are essentially your one-dimensional path integrators, and they might divide up in n-dimensional spaces somehow. And then you have this projection onto this thing, which is the readout part. And what is that? How do we understand that projection, that transformation? What would happen under different configurations of VCOs onto this two-dimensional canny thing that we have here? If I think of that as a readout, then it's fascinating. I mean, in some sense, perhaps this differential firing rate that I keep talking about between these phase clusters could just come naturally based on a set of VCOs that were spanning a different space. I don't know. It's just an idea, right? It's like if I didn't have these, if they were distributed in other ways, maybe I'd end up with a coding like this. So that this may be the reflection of a two-dimensional CAN with a three-dimensional VCO or n-dimensional VCO model. You might end up with something that looks like this.
Can I show you something?
Sure.
That might blow your mind with regards to this idea, which would be this little idea. So there's a pretty neat way to account for pretty much all the spatial cells that we do have. K cells, grid cells, different scale, border cells. What's actually not in here is also band cells, with the following idea. You have an array of VCOs. They're all different. And
where are you pointing? I didn't see where you're pointing.
this is the array of VCO, like
That,
that big array, the biggest
two-dimensional
part of the
basis.
figure,
Okay,
right? That's a big array.
Okay.
And so they're sorted here in like a 2D grid, just to denote sort of two different things. One is their preferred direction, and the other one is the slope of their speed modulation. If you sample perfect 60 degrees, cells that have the same phase and the same speed modulation in 60-degree separated directions, you get a grid. If you do that with a lower speed modulation, so you change the theta, right, of the theta cells, you get a different grid, right, larger grid. If you select cells from all possible directions at the same phase, you're going to get a cell that fires exactly in the middle, because that's where all of the oscillations will overlap to form a singular peak. If you select cells in only one direction, but at all possible scales, so to speak, then you get something like a border cell. If you select a set of cells, if you'd say you select only one of these or two of these, instead of a nice 60 degrees separated pair of three, you would get something like these other spatially periodic cells, like band cells. So a single array of these, right, depending on how you set up connectivity, can actually not just generate grid cells, but pretty much all of them. So it's in some sense it's sort of like a universal theory of spatial cells based on VCO arrays. No can require, right? This is just oscillatory interference.
I still have a basic misunderstanding of the VCO cells. I don't know how to describe it here. Let me try to phrase it. So I can imagine if I say here's a ring of cells that represent a preferred direction. So if I'm moving that direction, the activity in the cell moves around the ring, right? At maximum speed. Well, it's speed relative to.
Right.
So I imagine, okay, great. I've got a whole bunch of these VCO rings, if you will, one for each dimension, each direction. And if I'm not going in that direction exactly, but I'm going at a not orthogonal to it, but slightly off, I would expect that bump still to move, but not move as fast. Is that
right?
Correct?
That is exactly.
Leave this picture up here.
Okay, I'm sorry, because I wanted to show you exactly what you're talking about.
I want to ask you here. So what I don't understand is if I say, okay, I have a ring of cells, and let's say, is this the maximum rate here? Or is this maximum rate? Maybe this is maximum rate, right?
Yeah, it's higher speed modulation.
So basically I'm saying these cells, basically, this is a set of cells. We're going to move between these cells at the maximum rate when I'm moving. Well, it's going to be the velocity in this direction times the maximum rate, and that's how fast we'll move.
Yes.
Is this a different set of cells, or that it's got this preference, but it's always going to be slower, or is this the same set of cells here? It's just representing when it's moving slower.
No, there must be different cells because they're actually, going to be in a different phase, right? In different phases then, right? One of them is going to be faster and the other is going to be slower. And they've actually observed that there's different cells. So there's.
Well, I think about these as one-dimensional path integrators in some sense.
Mm-hmm.
I can have a set of cells going around the ring, and the correcting for path integration is that this same cells would either move faster or slower depending on the orientation of what I'm actually moving. But I wouldn't have multiple sets of cells. I wouldn't have one set for slow moving or the off direction, another one on direction. I don't need one set of cells. Why would I have a set of cells that only doesn't get from being marginally in that direction?
Because if you wanted to have a thing like these border cells, for example, or grids at different scales, then you need to sample from VCOs that move slower with respect to speed. So like the slope of the speed modulation must be different.
But so when I said the slope of the speed modulation different, is just
It's a different modules.
okay.
Right?
So it's not like. Would it be true then to say that just take this module?
Mhm.
Is it true to say that I'm going to move at some velocity in the preferred direction, and I will and it'll move at some rate? If I move it in a slightly non-preferred direction, it'll still move at just a slower rate.
Yes, that's
Okay.
a cosine law.
Okay, so to me, I misunderstood this diagram a little bit. This is not to say that these cells are not optimal for the direction it's moving.
No, they're exact, all
They
optimal.
are all optimal, they're just representing different scales.
Right.
This is to me, I didn't translate these pictures to scale. That's what they are.
Right.
They're saying this would be a large scale because it moves slower, so I have these big spaces.
Yes.
This would be a small scale.
Right. And you can see that by the fact that the two different grids here, the dorsal and the ventral grid, they sample in green and blue.
Yeah,
Right.
so
The
there's
green grid is tighter because it has a higher slope in the speed modulation.
so these are just more extreme versions of the dorsal and ventral. So, right?
You could think of it that way, yes.
Okay. And the dorsal and ventral, is that the 1.4 scale thing we're talking about typically?
Yeah, of course. That's an interesting question. Why does the speed modulation jump?
No,
with
not why,
Attractor?
but that's what we're talking about.
That's what we're talking about.
So,
The jumps and scale across the
in
dorsal
the
and
entorhinal cortex, they see a series of those. I don't know typically how many, 4 or something like that. Something like that.
Yeah,
So maybe like 4 of these could be typical of what we would see in the entorhinal cortex. And these are the different scales.
I think Moses said that there might be 5 or 6.
Okay, fine. But whatever.
I mean, it's fine. It's like
Yeah,
a
it's
finite
fine.
number.
So they're different scale. I was confused by that. Got it. So this is a set of vectors or a set of path integrators at one scale, another scale, another scale, another scale, another scale. Can we go through? I want to go through the picture again now slowly. You're saying here, if I sample across all the different scales, I get a border cell.
You're gonna get a border cell.
I'm not sure why. This I get, that I get. And this one's, they say, if I sample across all the different orientations, yeah, I don't believe that's what's going on up there, but you would get that, but I don't think that's right. It's just like throw it on the chart, what the hell, why not? Yeah. The other interesting thing is we talked about whether there's evidence that there's cells that sample across the different grid cell modules at different scales. And there really isn't much for that. Maybe it's true, but
It was originally proposed as a method to get to place cells, right? By looking for the overlap in grids.
well, that's one, yes. But in general, it doesn't occur. And well, the evidence we reviewed, as far as we know, we didn't see evidence.
There's also not strong evidence against it. It's just you don't look at it and find obvious connections.
You don't see the obvious connections. It kind of violates some sort of general rule we know about neural tissue, about spreads and what connections look like. But it's not impossible. So this one's a little suspect to me, this guy, although it's nice that it gives me words.
Can I make an argument for the place cells like that?
Sure.
It's known that place cells still update in the dark.
Yeah.
So you would get that if you had a grid cell based mechanism sort of into that.
Remember, you'd also get that if you learn the place cells associated with a particular location. Right.
Yes.
Right.
Right. Yeah. It gets.
There's a fundamental need to map sensory data with location that's independent of sensory. That's like a fundamental thing to model something. And so anybody who explains place cells without invoking sensory data it can't be right. It just can't be right.
Can I blow your mind again?
Sure. Well, I wouldn't go on this one. I was confused by it, but go ahead.
At least, I mean, I found this picture fascinating.
Well, it's nice to know.
Not that I believe it's all true, but it's beautiful, right? Yeah.
People are asking what this paper is.
Oh, that's the end of the Welday 2011 paper that I talked about before, the one that showed the VCO law, the cosine tuning, and the here. Hold on, I'll put it up.
And I'll get the
this
link
one
from you
here.
after too.
This paper. It's a Journal of Neuroscience paper 2011 by Welday et al. This one.
Thank you.
Okay, you're going to show one more thing. I know
Yeah, I wanted to show one more thing to blow your mind, which I think is a nice argument for a hybrid model also, which is this. So this is part of a presentation that Everett Moser gave. And he was talking about the weird thing that somehow, like often the angle of the orientation is 7.5 degrees with respect to the boundary in a square environment. And then he showed this cool thing.
Matt, we got your
No, I think it's close speaker.
So the fascinating, obviously, people can look this up if they want. There's the title up there. But the point is they found this fascinating grid cell here with a mean orientation of -13 degrees, which is like unheard of. And so it turns out it's not actually a mean grid, or it's not a grid at -13 degrees. It's actually two grids at 7.5 degrees. And what you see very nicely here from this picture, when you break this grid down, it's sort of like it bends around. It's really two grids in one, and it switches from one to the other. So suggesting that this grid is first anchored to one of the walls, and then when you cross sort of this diagonal, this grid is anchored to the other wall. So the whole well, the grid bends, right? It switches between.
I mean, it's the same cells changing their anchoring.
Right, their grid orientation changes, right? You can see the scale is preserved, but they start drawing lines to this, and it turns out that this very much looks like the grids are local, right? And sure, there's this nice, distance between them, sort of suggesting that might be some like network effects. So if you have a boundary anchor to one wall, you get the other cells sort of play along. But that doesn't go infinite, suggesting that the range of inhibition is limited, because this grid, when it gets anchored to the other wall, but it actually switches into a different.
Can we go back to the previous diagram?
Yeah, sure. Like this doesn't blow your mind? It blows mine. That's a major argument for a hybrid model, I think.
Well,
what's the connection from this to the hybrid model? This could be described without even talking about how grid cells.
No, it shows that you want a weak CAM.
Well, it doesn't even we can. It just says that you have an anchoring mechanism for any mechanism, any kind of cells. You have an anchoring mechanism, and then there are times of conflict where you have to go from one anchoring mechanism to another. I mean, that's just has to occur.
But why does that mean that there will be that the neighboring bumps will abide by the same grid? That only works. That only works with the CAN mechanism.
Yeah, of course.
Yeah.
So a CAN model that has to anchor.
Right, exactly.
All right, but I mean, it doesn't say a hybrid model. Anyway, can we go back to the previous picture?
Which one? You mean the Welday?
I mean, we've seen these pictures before of the grids getting distorted and have to re-anchor different parts of the room and so on.
Mm-hmm.
So the border cells here was, in that theory that the border cells are anchoring everything. In some sense, like, okay, the border cells are not going to set up my CAN thing as much as I can until another border comes along and interferes with it. The border require sampling across all these. Well, this says you can get border cells by doing it. Were there other models for getting border cells? Marcus, do you remember? Were there other proposals for how border cells came along?
No, I think a lot of them are more sensor driven. And I don't know. Usually, I think border cells are seen as more like they are coming kind of sensors helping drive border cells, which helps anchor the grid. I usually don't see this kind of thing.
Yeah, people typically treat border vector cells as sort of like an input to the system rather than an output. Right?
Border vector cells is different than even this, right?
Well, there's some border vector cells in the downward direction. It doesn't matter how long the boundary is. So that's why they call it vector cells.
Okay. Thanks.
All right.
Do we have to end this?
Yeah, I think we should sum up. We only went through like two of the results figures here, but that's all right. I guess I convinced you that it's possible to put these things together and that there's some...
we were convinced that a long time ago. So
Well, okay, all right. And the point is there's quite a few beneficial side effects from that. For one is that, in addition to all the oscillatory interference stuff, you now get more stability. You get to account for intracellular voltage dynamics, meaning you get essentially all of the benefits of both models. And some of the vulnerabilities of these individual models go away. It's still, I find it so obviously there's problems with the hybrid model. For one, the intrinsic theta reset mechanism that they have is very top down. It's just a heuristic, and they don't show how the VCO's connections are learned.
again, could that go away if we really assume a different role for place cells? Is that the thing we were talking earlier?
No, because then the place cells would have to do the same thing, right? You would still need to build a microcircuit that actually does the theta reset.
All right, I guess I need to study that.
So whether the reset gets done by grid cells or by place cells makes no difference with regard to the circuit that you're going to need to build in order to implement it. And they didn't do it. Right. The other one is, of course, that the interaction that they have between place cells and grid cells, which we think is important, is like rather crudely implemented. They just have this implementation where they sharpen the grid by having spatially selective excitation. That's not really like a circuit mechanism. And yeah, most of what I would like to see is like how are these VCO cell input learns, because there is an actual proposal for a learning rule, but I have yet to see somebody implement it.
Okay.
So I guess one could say, Russia, if you're listening, or Neil Burgess, if you're listening, I hope you can find the 30,000 model details that are missing. Our academic press might reward you or something like that. So there's lots of top-down model aspects that I think deserve fleshing out, even if the benefits of putting the models together is quite obvious. Yet at the same time, the model also has what I think is mechanical overkill. Like if you just look at the NVA model here, it's like a massive thing. I don't know, they have magnesium concentration in there that they don't do anything with. Yeah. So on the one hand, it's a very detailed model, but on the other hand, all of the stuff that is really mechanistically interesting, how it comes about, how it gets learned, and how it gets built dynamically is sort of put to the side. So it's a very awesome proof of concept that you can practically put these things together, but it's very engineered. And it would be nice to actually do we talked about the topography of this, right? So to actually do that in a topographical sheet, right? Which would then also get to how do these connectivities get learned. Right?
My summary from today is, this is for my benefit as much as anything, is that it's this idea that the VCOs are separate. They are the path integrators, and that the way we might think about grid cells is the readout of them, and that one level of indirection provides all kinds of interesting possibilities, and that could explain how we map from n dimension of VCOs to 2 dimensions of grid cells, and it might even explain the weird little stuff that's going on here. And my other walkway for me personally is I have to understand the whole cellular VCO mechanism better. I don't understand that. So I'm just speaking for myself.
Okay.
Okay, great. That was good.
Cool.
Excellent.
My pleasure. All right.